Hi, I'm Matthew Berck. I work for Araby Samuel in his lab in the physics department of Harvard. And I'm going to be talking about the olfactory system of the Drosophila larva, in particular its connectivity, as well as some experiments we've done on the physiology of that system. But before I talk about Drosophila larva itself, let's talk about olfaction. Olfaction is a very interesting problem in neuroscience of how an animal can recognize distinct olfactory cues and use that to drive innate and learned behaviors. And what's remarkable is that amongst both vertebrates and invertebrates, there are remarkable homologies within the first few layers of the olfactory system itself. Both have olfactory receptor neurons at the peripheries. In the mammal, it's in the olfactory epithelium, in the larva, in the fly, it's in the antenna. That then goes to a first relay processing center, the olfactory bulb and the antennal lobe. And then from the antennal lobe, you go to the brain, where you go to two different areas. In the adult, you go to a place called the mushroom body and the lateral horn. The mushroom body is thought to play a dominant role in learned behavior, and the lateral horn is supposed to be a dominant role in innate behavior. And there are analogous structures in the mouse brain as well, which would be your piriform cortex for learning and the amygdala. But even beyond the brain regions, you have very specific, very strong homologies within the first relay structure of olfactory processing. Both the mouse and the fly have glomerular organization, where you have neurons of the same type converging into the same area of the first relay. So neurons that display the same olfactory receptor olfactory receptor proteins, which have selective responses to odors, go to the same place. Then there are glomerular-specific projection neurons that leave and go to those two higher brain centers, and then the communication between glomeruli happens via mostly inhibitory neurons that are called local neurons. So the takeaway of this slide is that there are three main types of neurons, which you're going to see a lot of. Olfactory receptor neurons. There are local neurons that perform computation between glomeruli, and there are projection neurons that leave the first processing center. But we don't study the adult fly, we study the larva. And the fruit fly larva has what I would say are three main advantages for studying the larva. The first, which I excised from the talk for time, is that it has very easily quantifiable behavior. So you can look at the inputs of a sensory system, and you can look at the outputs of the larva, and it's a very easily quantifiable situation. Another thing that's very important for at least the last part of my talk is the fact that the larva is transparent, which means that you can do something like this. You can take GFP and express it in every single neuron in the larva, and you can see it without having to do any sort of dissection or anything like that. So this would be its central brain right here, and all of these little green things are its sensory systems, also motor neurons. It does not have antennae, but it has a dorsal organ, which is at its head, which is where all of its olfactory receptors are. The other great thing about the Drosophila larva is, whereas the adult fly, the main reason to study the adult fly is for numerical simplicity over, say, a mouse, in the sense, the fruit fly larva takes numerical simplicity to the next level. You don't have, the adult has about 1,200, 1,300 olfactory receptor neurons. The larva has 21. And the 21 olfactory receptor neurons in the larva are all unique. So in a sense, if you want to define glomeruli, they all go to different glomeruli. They all have specific projection neurons that are one-to-one, and but they still have this very interesting communication between glomeruli via local neurons. And that's what we wanted to see. We wanted to look at how olfactory information is processed in this first relay. And we expected to see very interesting ways you have gain control and such. But what we actually found, which is even more interesting, is that this is half of the story, and there's an entirely separate pathway in the Drosophila larva olfactory system. The way that we did this is we have this very great collaboration with Albert Cardona at Familiar Farm, who's very close to here. And he has a serial section electron microscopy data set of the entire larval brain. There are about 10,000 neurons in the larval brain. He wants to get the connectome of the entire thing. He allowed us to look at the connectome of just the olfactory system itself. And so the numerical simplicity allows us to actually have the ability to get that connectome. And we used his software, which he calls Collaborative Annotation Tool for Massive Amounts of Image Data, or CATNAME. And so what we did is we reconstructed every neuron that innervates the antennal lobe. And what's great about this data set is it's of sufficiently high quality that you can even see synapses between neurons. So you can get not only the structure of the neurons, you can also get the connectivity between all the neurons in the antennal, the synaptic connectivity between the neurons in the antennal lobe. So the tour de force result is the entire connectome of the antennal lobe, which would be this. We have two of them. There are two antennal lobes. But this is difficult to understand, so let's look at some skeletons first. So yes, the larva has 21 olfactory receptor neurons. Yes, it has 21 one-to-one projection neurons that go to the mushroom body, which is here, and the lateral horn, which is here. And yes, it does have local neurons that process the center. But what's interesting is that there are two very distinct types of local neurons, which is entirely unexpected. One set of local neurons is for processing the uniglomerular projection neurons. The other set of local neurons is for processing these multiglomerular projection neurons that are not glomerular specific. And there are two different circuitry motifs that come out. So in a sense, you have two parallel circuits in the antennal lobe. The one circuit being where you have two types of local neurons, which we call broccoli neurons, because I think they look like a head of broccoli, and picky local neurons that do not innervate the entire antennal lobe. And what you can see is that these panglomerular broccoli neurons are for processing one-to-one projection neurons, whereas these very specific local neurons are for processing these highly selective and also specific, although not as specific as the one-to-one projection neurons. They're highly selective and stereotyped from side to side, multiglomerular projection neurons. The double-headed arrows means that you have reciprocal connections. So the way that the panglomerular neurons process this information is they provide inhibitory inputs to every single ORN, every single PN. And they receive information from every single ORN, every single PN. So these things are doing some sort of normalization, which expands the dynamic range of how these ORNs can respond, so it can respond to multiple concentrations of odor. What these picky neurons are doing and these multiglomerular projection neurons are doing is something entirely different. What they're doing is they are performing very highly selective inhibitory processing. You have direct connections via these green lines, but there's also this very strong lateral inhibition motifs from certain ORNs and other projection neurons, as well as this sort of mixed motif where you have direct and a few forward connections. But what's very interesting is that these things seem to be segmenting odor space. So you can see, we don't know what 49A does, but we know what the other things respond to. The neurons that are aliphatic for this particular multiglomerular projection neuron are forming this direct and feed-forward connection to this multiglomerular projection neuron, whereas the aromatic ones are direct. And now that's pretty much the summary of what we found in the thing. I can go into details and talk about it for hours, but I won't. What I'm going to do is I'm going to talk about the way that we measure these neurons that people in the lab have developed and that I've started using myself, which is via these microfluidics. So the way that we can measure, the great thing about a larva, again, is that it's transparent, which means that we can measure the response of their neurons using a green fluorescence protein that fluoresces when the neuron is active. It's a calcium indicator. Calcium flows into the cell when the neuron is active. And we can look at their ORNs. This is in the dorsal organ itself. The local neurons, projection neurons, everything. And what's great, let's restart that movie, is that we can look at the response to these things using calcium imaging. So this one, we're going to give it increasing concentrations of, in this case, 1-pentanol. You can see that we have one ORN respond, and suddenly, oh, wait, there's a little bit more. And now we have like 5 neurons responding at the same time. So as we increase the concentration, more and more ORNs are active. You can also do this in the antenna lobe proper, which you can see that there's a lot of spontaneous activity, which is something that we're very interested in. But you can see we gave it 4 different odors, and we give it another prolonged odor very soon, boom, and it's going to stay on. So we can actually segment the antenna lobe fairly easily by just looking at correlations between adjacent pixels. So you can see what glomeruli are active and when. And since the glomeruli have very stereotyped positions, we can actually identify them. You can see that there's a lot of very interesting things happening here, but the one I want to point out is that in this neuron that never responds, the spontaneous activity is severely is dampened when you give it a prolonged odor response. We can also give it a random stimulus, and from presentation to presentation to presentation to presentation is practically the same every time, which means we can probably calculate a transfer function, a filter that will take you from stimulus to the fluorescence, which what you can do, and it does a pretty good job, especially if you give it a nonlinear component to the filter. This is another random stimulus, but using a filter from another stimulus, and then this is a periodic response. So I'd like to thank my advisor, Eric Samuel, who did a lot of the development of my microfluidic device, as well as Christopher, who did a lot of the genetics, and Albert Cardona in his lab for the EM data set.
I was wondering, there's a few neurons that are distinct and one-to-one, what does it say about the robustness and plasticity of the system?
I'm not sure what your question. What do you mean by robust? There's 21 neurons. It's very robust if you look at the numbers of synapses between these neurons. There are hundreds, which is very different than, say, the adult, where a single ORN is connecting to a projection neuron 20 times or so, and you have all these things. These neurons, what they'll do is they'll make 120 synapses to the same neuron. So there's a lot of this, and there's a lot of noise in the connectivity. You'll get 2 synapses for all over the place, but you'll get these very strong connections to neurons that are on the order of 100 synapses. So it's fairly a robust system.
If you would take out one neuron, let's say.
Oh, you're saying you take out one neuron when you lose. Not all ORNs are created equal, so you can get rid of, if you get rid of certain ORNs, they won't be able to do taxis at all. And they're very important ORNs, especially, say, OR42A, which senses ethyl acetate, which is something that is given off by like rotting fruit. So you can look at, so if you knock out that neuron, they can't, the taxis is gone, right? These animals are only alive for 4 days, so their systems are not particularly robust, because they don't have to live for very long. They just need to eat a lot. So, and since they're born in food, they just eat all the time. Right, so it's not a very robust system in and of itself. You can get rid of like unimportant ORNs. There's certain ORNs that don't seem to drive behavior at all. We've done optogenetics activation of these neurons individually, and you can see that half the neurons don't drive taxis on their own. They're supplementing it in some way, in probably for like by distinguishing between odors is probably what they're used for. And there's also some very aversive ones they're dangerous things that the larva need to avoid, and so there's certain olfaction ones that are aversive as well. But you can get rid of those and no longer do aversive stuff.
And is this different in higher animals? I mean, I would imagine that have more redundant systems.
Oh yeah, for sure. The adult fly has 50 types of ORNs, 40 copies, 20, 30 copies of each ORN. That kind of thing.
So does every single larva have the same 21 different ORNs?
Yes, that's very serious.
And we know how that works? I mean, I asked Arabi this question. He said sometimes, yeah. So, I mean, that's a separate question from how it functions as a neural system, but it's sort of interesting that you create 21 cells, each one has one specific gene, and all the others are somehow turned off, and you make sure you have one cell of each.
Right, yes. I mean, I don't know how to, it's a very, I don't know what do you mean it sometimes works? I mean, they all have the same.
Right, no. No, I mean sometimes works. I mean just that you have to create a system where all those genes are in every cell or in every neural stem cell. And yet you create 21 cells and each one has a specific ORN and once you've used it once you don't get to use it again, and so that's an interesting program.
It's a very interesting problem.
Right, but there's nothing to do with your work.
Right, that's exactly right. It is a very interesting problem. I entirely agree with you.
Huh?
The heat.
I can't hear what you're saying.
The heat also an interesting problem.
One more question. I don't know how this applies to the fly olfaction system, but I know that in at least probably mammals, there's been some work trying to understand that certain olfactants have an inhibitory effect on the neurons. Are you able to, do you account for that in your modeling or in the way that you are measuring these things? Can you
Yes, so there is an effect of if you present an odor, which we're trying to get around by. It's possible that there's an effect between ORNs. So if you excite one, there's an electrical inhibition on an adjacent one, and that seems to actually be true. You can get around that by using only single active ORNs and such, but there's also the possibility that you're blocking a pocket so you can get some sort of loss of spontaneous activity. You see all of that. It's not something that we haven't started modeling any of this yet, so we haven't accounted for it. But you definitely see it with the microfluidic. You'll see huge, sometimes large, sometimes small drops in activity, neural activity, just by using a mixture of odors as opposed to using one odor.
Right. Let's think.